welcome back to the Assay Guidance Workshop on DNA-Encoded Libraries for Lead Discovery. I'm going to kickstart day 2 now. Thank you very much, whoever is joining us again today. Hopefully you enjoyed day 1, and we hope you will have a interesting and an exciting day 2 as well. Okay, so before I start, I'm going to show you again our Twitter handle, which is at Assay Guidance. Please feel free to follow us and tweet about this event using hashtag AGMDEL2021. Okay, so if you didn't join yesterday, I'll introduce myself again. I am Sarine Markossian. I'm the editor-in-chief of the Assay Guidance Manual and the lead of the AGM program here at the National Center for Advancing Translational Sciences, National Institutes of Health. Okay, so before we start the program today and learn all about cool stuff about DELs, I would like to tell you a little bit about our program, our AGM program. I will not take too much time. Hopefully within 10 minutes or so, I'll send a message to you all so that you can appreciate all the resources we are sharing with you that might be helpful in your research. So, and let you know also that we are really trying to set the standard for rigor in translational science. Okay, so the outline of my talk I'll start first talking about the importance of rigor in translational science, introduce you to the Assay Guidance Manual Program and its main components, including the eBook and the workshop and the trainings that we run. And then we'll delve in today's agenda and tell you about all the exciting day that we're going to have today. In 2019, Translation Together, which is a international collaborative effort to advance translational innovation, and NCATS is part of Translation Together. So the members of this group published a manuscript in 2019 that defined the characteristics of a translational scientist. And those characteristics include being a team player, a rigorous researcher, a boundary crosser, a process innovator, a domain expert, a skilled communicator, and a systems thinker. What I would like to focus today is being a rigorous researcher. And what that means is to conduct research at the highest levels of rigor and transparency and possess strong statistical analysis skills, as well as design research projects to maximize reproducibility. And this is a very important term because there is a crisis of irreproducibility in basic and preclinical research. A manuscript published by Friedman et al. Showed that the cumulative prevalence of irreproducible preclinical research exceeds 50%. Now, this results in approximately about $28 billion spent on preclinical research per year that is not reproducible. The categories of preclinical irreproducibility include biological reagents and reference materials, study design, data analysis and reporting, and laboratory protocols. The Assay Guidance Manual Program is designed to address this problem. So the AGM is a disease-agnostic program that addresses this crisis in basic and preclinical research by setting the standard for rigor in translational science. What our program does, it sets best practices and standards for assay development, implementation, and analysis. And more information about our program can be found in a manuscript published by Cousins et al. In Clinical Translational Science in 2018. You'll learn more in this manuscript about the history of the program, all of its components. So have a look when you get a chance. Now, I'll tell you a little bit more about our program. The main component of our program is the manual itself. It's made of 55 chapters and over 1,300 printed pages, all downloadable in a PDF format. It's free and publicly available resource since 2005, currently accessible as an ebook on the NLM and CBI bookshelf. Because it's an ebook, it is actually dynamic and has continued renewal and expansion of content. It is widely accessed, has more than 30,000 views per month since 2017, actually, and we see similar rates this year as well by scientists from pharma, biotech, government, and academia research laboratories alike. We are supported by 34 editors with extensive experience in preclinical assay development and drug discovery. And we have a Creative Commons license, which permits copying, transmission, and adaptation of the work, provided original work is properly cited and not used for commercial purposes. The manual is composed of multiple sections, and I don't want to go into too much into this, but some of the sections have more chapters than others. So the heavier chapters, sections include biochemical assays, cell-based assays, and assay validation operations and quality control. And I would like you to know that we keep expanding. So we add more chapters into these sections. We're also expanding our sections. So for example, we're really excited to let you know that we will bring in new chapters into a new section on antibody therapeutics in the upcoming years. So, and also we're always bringing in new chapters. So if you're interested, look through our manual, and I really hope you find our manual helpful. What I do want to share as well is that our manual is a very popular resource. So the AGM itself has been accessed about 100,000 times per quarter since 2017. As you can see clearly in this graph, monthly rate of access varies between 30,000 to 45,000 times per month, depending on the month. The Assay Guidance Manual is also accessed by 131 countries. So we did a study and we saw that during the month of April 2017, the AGM was accessed 40,000 times by reader in 131 countries. So what you're looking at is a heat map, and it's showing the amount of access per country ranging from low dark blue to high red. So it's really exciting to see that 131 countries are accessing our manual. What's also exciting is that we've been running a classical assay guidance workshops for high throughput screening and lead discovery. This has been our classical workshops that we've been running since 2015. And we've run so many of those, and they've been really popular. And the last one we ran was in November 2020, and that one was virtual. We made all the recordings of this workshop available online, and you can find these recordings at this website. And I really highly encourage people to go and check this, the recordings of this workshop, because this workshop really covers a lot. Like the scope of this workshop is big, goes from all the way assay development for cell and biochemical assays to data analysis, new modalities. So there's a lot of really interesting talks in this website and this workshop. I do want to mention that we are going to make all the talks of today's and yesterday's, so the whole DILI workshops recordings also available online for you, and we will email everyone who's registered with that website and the links of those videos. Okay, so future workshops. We will keep running our assay guidance workshops for high throughput screening and lead discovery. So we have two upcoming, one in 2022. The first one is going to be as a short course at SLAS 2022 on February 5th in Boston. And this, we'll see if the pandemic will allow that being in an in-person one. And the second one, we're hoping to run in person at the NIH main campus on October 2022 in Bethesda, Maryland. There are multiple upcoming new topic-focused workshops and symposia. The first one is the Assay Guidance Workshop for Cell-Based Assays and Lead Discovery, which will be held virtually on November 10 and 11, 2021. Registration of that will open soon. In case you're interested, please stay connected with us. And then the AGM symposium, we hope to run an AGM symposium on antiviral drug discovery in 2022. And also we hope to run an AGM workshop on antibody therapeutics in the next year or two. So again, connect with us to stay tuned about upcoming workshops and symposia. There's multiple ways you can connect with us, including through Twitter. Use the handle @Assay Guidance, on LinkedIn. This is our LinkedIn group. And then also we post all our events on NCATS website, as well as the NCATS AGM website here. With this, I would like to acknowledge NCATS leadership, including for their support of this really exciting program, including Johnny Rutter, who's the acting director of NCATS, Anton Semyonov, who's the scientific director of the Division of Preclinical Innovation at NCATS. As you saw, Anton gave the opening remarks of this workshop yesterday, and he's been really an active member of the organizing committee, helps us organize this workshop, and has been very supportive to the AGM program. So thank you very much, Anton. And then also a big thank you to Matt Hall, who's the director of early translation branch at NCATS, and the AGM program is part of that branch. So, and he's been extremely supportive of this program and of this workshop as well. So thank you, Matt, so much for all the support. A huge thank you to the AGM team, Abigail Grossman, a huge thank you to Abigail. She's been driving force of this program, and as you've noticed that she's absolutely instrumental for this workshop running smoothly. So thank you so much, Abigail, and for everyone in the Abigail's colleague in the IT department as well for making this workshop happen. Next is Sita. A huge thank you to Sita Sitampalam. He's actually the founder of this manual, and he was the editor-in-chief of the manual prior to me. So a huge thank you to Sita and Kyle Brimakombe, who's a program manager at ETB, and he's been also extremely helpful with this workshop, and many others as well. A special thanks to all Assay Guidance Manual editors and Assay Guidance Workshop organizers and speakers. Okay, so with the last 2 minutes, I'm going to jump in and show you the program of today. So we'll continue session 2 in a few minutes on selection, and then we'll have another session on data analysis, and that will have 3 talks. We'll have a break at 2:30, and then resume session 3 at 3 o'clock. In the afternoon at 3:40, we'll have a panel discussion on emerging trends and technological advances. This panel, we have really exciting short talks in this panel, and we're going to have a very engaging discussion. So I'm really excited about this panel. Do not miss it. And also, I would like to mention that throughout the whole meeting, throughout the talks, you can plug in your questions through Q&A, as we did yesterday. I was extremely happy to see the level of engagement with the questions yesterday. So let's keep it up. Hopefully you guys are, asking all the questions that you have, so that we can help you understand about DELS better. And we are going to do our best to answer all of your questions. I'm going to mention one more thing, and that is we're going to ask for your feedback after this workshop, and we would love to hear what we are doing right, what we are doing wrong, so we can improve these workshops and hopefully run many more workshops about DELS in the future. So your feedback will be extremely valuable. I highly recommend you to fill this feedback when we send it to you. So we are going to send it to you through email after the workshop.